My name is Jonna. I'm a graduate student at Duke. I study neurobiology and I work with bird brains. So I'm going to tell you a little bit about music, a little bit about rhythm, and a little bit about the brain across the talk. And again, thanks for coming, at least to listen to a little bit about this stuff. So keep a good eye on this screen. I know it's small, but a lot of the stuff that I'm going to show you is visually really crazy and conceptually even crazier. So I would really like you guys to be able to see what I show you up here and to actually also to listen to the clips that I'm about to play. So this that you're seeing up here is a picture of a single neuron. That neuron is just chilling on a plate. It's been genetically engineered to look red. And the cool thing about neurons is that, for example, a single one could send or receive, give and take, 10,000 inputs and outputs. So one single cell can send and receive 10,000 messages around that amount. But this gets really complicated when you keep in mind that your brain doesn't just have 10, 20, 100, 1,000, a million neurons. It has somewhere on the order of 200 billion to 300 billion for the average adult brain. It has about 300 billion neurons maybe. So once you take into account that each of those neurons can make about 10,000 connections, we quickly get into the trillions and the hundreds of trillions of connections throughout your entire brain. And that makes understanding the brain extremely challenging, and this is why it remains to date probably one of the biggest mysteries in human existence. So this is another single neuron. It's been genetically engineered to look green, and I'm going to show you what you would hear if you could listen in to what that single neuron is telling its neighbors. And so it might sound something like this. So it kind of just sounds like non-structured crackling and popping, right? But in reality, every single little pop you hear is an action potential that the neuron is sending. So when I say action potential, what I mean are these little electrical impulses that neurons can send that are essentially a yes or a no, or a 1 or a 0. And they can send these to their thousands of neighbors, and they can receive 1s or 0s from another thousands of neighbors. So this makes it really intense, the amount of computations that even one neuron has to do. And if I showed you the voltage trace, that is the electricity coursing through this neuron while it sends messages like this, it might look something like this, where you have these sharp increases in voltage that are super small, super quick, and those are these action potentials that I'm talking about. Now, like I was saying, neurons don't exactly exist in isolation. You don't just study one neuron. You don't just study a handful of neurons. There's billions of them. So what I'm about to show you is this really kind of mind-blowing technique that was developed about 2 years ago in Stanford, where you can take a piece of an animal's brain and then you can make it essentially transparent. But you can keep labels on things, specific subsets of neurons that exist within that piece of tissue. So what we're going to see is that a little cubic millimeter of tissue has thousands upon thousands of neurons that are all organized almost in an architectural fashion, and they could be doing really complicated things. So as we look through the tissue, you can see, so this is actually a piece of what's called the cortex of the mouse, where a lot of the higher cognition happens. And you can see that a bunch of these neurons are organized almost in layers. There's particular motifs to them. They look like they're structured for sure. And these are only a subset of the neurons in the mouse brain. So clearly, if we want to understand the brain, we can't just look at one neuron, not a handful of neurons. We have to keep in mind that neurons as a population are sending messages that we really need to care about, and that's how we might really start getting at, so like, what do brain regions do? What do pieces of your brain do? What does your brain do as a whole? So the two ways that I think are really useful to think about large groups of neurons doing things are in tempo and rhythm. And I mean, these are two of many, many, many ways to think about this, but I'm going to be talking about this today specifically. And the first one I want to talk about is tempo. So I want to raise the question about how do we as human beings keep a sense of time? Because if you think about it, it sounds like a very simple question, but it's actually really, really intense and intricate. When you think about it, you can keep time at sub-second scales all the way up to memories that you have from decades ago and everything in between. So your brain has to have some pretty damn sophisticated hardware to be able to keep all those parallel time streams somehow commingling in your brain. So today what I'm actually really going to be talking about is one particular theory underlying this idea of like, how do we keep time? How do we keep a perception of time? And this is called the metronome theory, or at least I'm calling it the metronome theory because it's a convenient name for today. And this is the idea that in some parts of your brain, there are structures of neurons that can somehow represent time in a uniform fashion. So that is, you have parts of your brain that can keep a regular sense of time upon which you can structure behaviors, thoughts, sensations, and everything else, and keep kind of a temporal reference to them. So it's actually been really hard to test theories like this and like anything else that involves the perception of time, but one of the really cool model organisms that we're able to use to study this is actually the songbird. So that's what I study. And the idea is that birds themselves, birds of song, have to be able to create these behaviors that are extremely tight temporally, extremely elegant acoustically, and sometimes they can even switch timing throughout the song, and they perform them with almost this mathematical accuracy that's just crazy. So the other reason that we study birds is that if I were to go into the brain of a human, so for example, in this case, this is my favorite drummer, Tony Royster Jr., and if I were to go into his motor cortex and to ask one neuron, what do you care about, neuron? That neuron might care about a bunch of things. So a variety of movements, a variety of actions can be encoded at a single neuron. That neuron can be responsible for a lot of it. And similarly, if we go into the brain of a mouse, which is one of the most common organisms that we use in labs to study stuff, you might find the same issue where if I go to the motor cortex of that neuron, of that mouse, one single neuron can be involved in a plethora of activities. The cool thing about birds is that if I ask the same question and I just go into one region of the bird brain, not only do you find that that region is associated with one single behavior, you actually find that a whole network of regions is just encoding this one single behavior, which simplifies this problem of how do I go from what a neuron is doing to what the animal is actually performing as a behavior. And it makes it a lot easier, and it's extremely useful for neuroscientists to actually have this kind of simplicity in their models. And to illustrate how crazy this can get, what I'm going to show you is a computer-generated sound bite where the computer is essentially generating what we call a random walk with some constraints, and that sounds something like this. It didn't. So that was generated by a computer. And what I'm showing you is a visual representation of that that's called a spectrogram, which is essentially throughout the frequencies, how much power, how much volume is being ascribed to each frequency. And it's just a useful way to visualize sounds, essentially. But what's really cool is you can feed this sound to a young songbird. So in this case, we're talking about a canary. And when he's still kind of a juvenile, a young bird, he can learn this pretty damn well. So when you look at the spectrogram of the bird, he was actually able to copy that almost perfectly to the millisecond level. And he was able to vary his tempo through the song. He was able to copy the note structure. He was able to copy the whole thing essentially perfectly. And this is really amazing. And so that's why we study these guys and why they're so useful for us to learn about how does the brain encode time, how does it chain together behaviors, how does it control motor sequences so elegantly. But I need to introduce a recent advance in neurotech that allows us to really tackle these questions, which is the idea here is that you have a part of your brain that's a metronome. The metronome is distributed amongst many neurons so that they're firing, for example, in a sequence and going pop, 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 but each tick is a different neuron. This means that you have to somehow be able to look at many neurons and not just 1, 2, 3, 20, right? It has to be way more than that. So to do this, there's been a lot of work put into turning neurons into optical sensors of their own activity. In other words, if I'm a neuron and I've been genetically edited with this technology, now when I become active, I also become fluorescent or bright as a function of my activity. So the more active I am, the brighter I am, right? So we can combine this with modern advances in microscopy. So this is called the 2-photon microscope. It's an expensive piece of gear, and it's really big. And you can put an animal under it, right? You can genetically engineer its neurons so that they are these optical sensors of their own activity. And now this microscope can look into these brain areas and see what all those neurons are doing. And so what this really allows us to do is that now instead of looking at this small pocket of neurons with an electrode, which is what used to be done, right? You used to be just placing in a metal rod essentially into the brain and hearing these recordings like the sound blips that I was showing you in the beginning from a handful of neurons, from a very small group of neurons. But with this, you're able to actually get a lay of the land and get an idea of what all the neurons are doing because you're literally looking at their activity. So I'm going to show you what this looks like because it is actually freaking crazy. So this is a mouse. Its cortex has been genetically edited to have this optical sensor. And what you do okay, this sounds a little gnarly, but you can remove part of its skull, and then you can put in a window, a literal piece of glass, onto its skull. Now you can lower that same microscope, fix the mouse under that device, and look with your eyes at the actual activity that these neurons are, eliciting over time, or like showing over time. So what you're seeing here, the big black branches are blood vessels, and we're gonna take a little bit of a tour. So the flashes that you see, they're very small. Those are actually individual neurons flashing in the brain. We can take 4 random areas, zoom in, and now you can see the shape of the neurons, and you can see that there's a lot of them. So there's probably upwards from 10,000 neurons being recorded from here. Complex algorithms can be used to isolate those neurons, show you their activity over time, and now we're not talking about looking at 10 neurons or 20 neurons or 30 neurons. We're talking about looking at 20,000 neurons together at the same time across a huge, expansive cortex or any brain tissue that can really tell you how is the whole brain working together as opposed to how is 1, 2, or smaller numbers of neurons working together. So this is still not quite what we need for birds, because can you imagine that you grab a bird and you just kind of chuck it under one of these huge devices? First of all, it has to stay put, right? So if I'm a bird and I'm like, I need to be under this microscope, but I'm never going to be happy, and so I'm never going to sing, because birds usually sing where they're chill and they're happy and they're comfortable. So that's not going to work. So a lot of work has been put into somehow making these guys smaller, making these microscopes smaller. And this is a bare bones version of the microscope I showed you before that might be worth upwards from $500,000 is now something that you can make with a couple $100 in your house, pretty much. It's much less resolution. It can cover less neurons. But what you get are videos that start to look a little bit more like this. So if you look closely, yeah, it's a much smaller space, but you can still see a lot of neurons. Actually, you can kind of make out their shapes, right, their connections as they come on. And there's a lot of neurons in this field still. So this is extremely useful because now what I can do is I can get my bird, I can genetically edit the neurons in the brain region that I think serves as the metronome so that they have this optical sensor. Then I can lower one of these little microscopes onto that area, implant it there permanently, and now I have access to the metronome neurons, and I can see what the hell they're doing as the bird sings and as the bird does different things. And so this is what that looks like. This is a picture from my lab where the bird is able to, it has a microscope on its back, and that microscope is pointing down on this metronome region. And this technique is called calcium imaging. So now that the bird is able to sing normally and is more comfortable, we can actually give this theory more of a test. And I'm going to run you through what is pretty much seen when you do this. So this is another spectrogram. This is the song of the bird, pretty much. It's about less than 1 second in length, so it's pretty short. And what I'm going to show you is what each neuron was doing during this 1 second song. So the neurons are going to be down here, and there's going to be an orange tick mark for the activity of each neuron and when it fell in time. And then they are sorted through when they fell in the song. So the earliest neurons will be plotted first, and then the latest neurons will be plotted next. And what you get, for example, for the first neuron, it comes on the beginning, but once you actually plot the whole population, what you get is this really nice, almost uniform representation of time across the entire song. And if the bird were to sing 5,000 more times, you would still see something very, very similar to this, where the same neurons are stuck to the same points in time, creating this like uniform metronome that can really cover the expanse of the entire song. So this is pretty good. It suggests that a theory like this could have some plausibility to it, but it's still not enough because it's just an observation, and we need to really start figuring out causation. And this is not enough to figure out causation. So can we mess with the metronome sometimes? And to introduce you to this, I need to also talk about another thing that's really cool about neurons, which is their temperature sensitivity. So this is a recording from a neuron from the same area, this metronome area, and you see that it has a very steady firing rate. Every tick mark you see is an action potential of those electrical impulses I was talking about before. And this is just at room temperature, for example. This is on a live bird. You're recording from one neuron. Now, if I slowly lower the temperature of this neuron, what begins to happen is that you can see how these action potentials, these firings, start spacing out, and as I cool it more and more, they space out more and more. So there's a pretty clear prediction to be made here. If I can slow down the metronome, I expect certain things to happen in the behavior that relies on the metronome in the first place. So what can happen, and this was done by Michael Long over at NYU, is that they implant a cooling device into these areas of relevance, and then they can focally lower the temperature of the neurons in that area for very controlled amounts and amounts of time too. And what you see is mind-blowing. So this is the control song. This is the song of a bird that's just singing in a normal temperature in a normal room. And as you slowly lower the temperature, what you see is you can get a stretch, a dilation of the song. It stretches a little bit. And as you cool it more and more and more, you start to get more considerable dilations of time for the behavior. And if you were to compare this last song, which is pretty long, to an artificially stretched version of the song, they look exactly the same. So it's not that its specific parts of the song are being spaced out differently or anything. You're literally grabbing the metronome and stretching the whole song by the same amount. And this guy is kind of crazy, so he took it one step further, and he collaborated with neurosurgeons at NYU that were doing deep brain surgeries that involved, like removing a tumor or dealing with epilepsy or something like this to implant an electrode. But they happened to be happening near where the speech centers of the brain were. So at this point, what he could do is he could sneak in with his grad student and just plant down a cooling device before the actual surgery and saying, okay, let me test my metronome theory on the speech center of the brain, which is Broca's area that a lot of you have probably heard about. And what you get is something that sounds like this. So this is the control sequence that the person was asked to speak. It's the days of the week.
Monday, Tuesday, Wednesday, Thursday, Friday.
Here's a cooled sequence.
Monday, Tuesday, Wednesday, Thursday, Friday.
So it's subtle, right? But what you can see is that essentially this person is not able to keep up with the tempo that was given to her to say the sequence over. In other words, they gave her this. She feels like she really wants to match it, and she can't. Yeah. So she can perceive her failure at matching the tempo by saying the days of the week. Yeah. So this is wild. And it's worth mentioning that sometimes, depending on where you are in the brain, you can also get it to speed up while still cooling the brain, which kind of speaks to the complexities of the circuits that exist there in the first place. It's not that straightforward. It's not like I can just dilate time by cooling down the parts of your brain that perceive time, right? So what I want to do now is change gears from tempo to rhythm, which is a completely different deal now. What I'm going to show you right now is a patient that has Parkinson's disease, and it's pretty late on, and they have what's called freezing gait. So they have really a lot of trouble initiating this behavior of walking, pretty much. And you can see that it's a big struggle. But you take the same person that literally cannot walk, and you put them on a bike, and then they're like, okay, see you later. Like, I'm going to go. And then if I let this video go longer, he actually turns and steps away from the venue that he's in, and then turns around and comes back, dismounts the bike, and then begins to not be able to walk again. You can take this another step further, and this is another example where this patient also has Parkinson's disease, and he has trouble with the same freezing gait. He has trouble getting here, but you can engage another kind of behavior that's also somehow intact by all accounts. So somebody painted stairs on the floor. So this patient has trouble getting to the stairs, but then once he actually gets to the stairs, he's practiced this already, he can engage in the illusion that there's stairs there, and he can kind of engage another motor program that allows him to function normally, despite the fact that he can never do this if there weren't an illusion to engage that motor program. So what this has done is this has led to the idea that there's these things called central pattern generators that are essentially little rhythmic circuits located all over your spinal cord and into the more deep areas of your brain that can function in isolation of a lot of other parts of your brain. And to really, really prove this next video is a little bit also tricky, but we had to do some other experiments. So these are experiments back from 1972 where a cat is on a treadmill. And you can see that this cat is able to walk pretty damn normally. In fact, if you speed up the treadmill, the cat can keep up and switch its gait from just a walk to almost like a trot and then a gallop, and it can keep up with this. The punchline to this is that that cat is missing a brain. It only has a spinal cord. And the way that, okay, this is really important because research like this is necessary to really get at these core mechanisms and to help us understand. But what is happening is that he's being held up by two cables, and then a stimulating electrode is in his spinal cord and is triggering at a faster and faster rate throughout this video. And that only increasing the frequency of the stimulation can get that cat to go from walking smoothly to trotting smoothly to almost galloping. So you can engage different behaviors with a linear increase in stimulation frequency, which is a really kind of core finding of these central pattern generators. They're not just flat out linear increases on some behavior. They can engage different behaviors. You can even go, in a lizard, you could go from walking to swimming, just from increasing the tempo of the stimulation in the spine. And then another part of our insight into these ideas of these like isolated rhythmic neuronal activities came from crustaceans, which was really weird. But fortunately, Mother Nature reuses a lot of the principles of these circuits all throughout the animal kingdom. So what they did was they went into a lobster's stomach, and there are muscles there that contract rhythmically. And so they lowered cables and they touched each muscle that contracts rhythmically. And what you get is something like this. Each color here that I'm showing you is a different muscle that's being recorded from, because you can also record electrical activity from muscles. This is really important. These are not the neurons. These are the actual muscles and their electrical activity. And then as you go through time, you see that there's very rhythmic cyclical activity through them. Then somebody asked, okay, well, how are these being generated? Can we kind of prove a little bit of these concepts, test a little bit of these concepts of central pattern generators in here as well? So you can, again, you can grab this crustacean, in this case a lobster, and you can isolate its nervous system from the rest of the body. So you can remove only the neurons and the nervous system and forget about the muscles, forget about the body, forget about everything else, and put it on a plate. And now you can put in an electrode and touch in on the neurons that control the muscles that I was showing you before. And then this is what it actually looks like. These are the neurons, and this is just on a plate. It's stretched out. The little blobs that you see in the center are neurons. You can record from the neurons, and what you get is that those neurons' electrical activity mimics, because it causes, the actual activity in the muscles itself. So you can find these rhythmic patterns without actually needing to interact with the world at all. These are just intrinsically generated by the neurons through these mechanisms that have been heavily studied throughout time. You can take this another step further, and how many people have heard about stem cells being able to turn into a variety of different cells by a show of hands? Right, it's a very popular concept, right? So somebody can put stem cells on a dish. It's just a dish. You can now, with different molecular players, you can turn these guys into many things. You can also turn them into spinal cord-like neurons. And so this is what you're seeing here. Every stem cell that's on this dish has produced what's called the neurosphere, so a clump of neurons, essentially. And these neurospheres can be played with to actually become more of an ecosystem as opposed to just a homogeneous blob of one single type of neuron. So that's what you're seeing here. And what you see is that just on a dish, just from one single stem cell, these guys start to pulsate rhythmically as well. And these pretty much just came from one cell. and they pulsate rhythmically, we can actually go back to this other technique that I was talking to you about, calcium imaging, where you can optically look at what these neurons are doing. And on a dish, these guys begin to pulsate rhythmically too. So it's a pretty relatively stable rhythm that they display. And this came from essentially one cell, which was the stem cell that produced these. And it was only played with molecularly to become this ecosystem that's very spinal cord-like. And this idea of central pattern generators has not remained confined to the field of neuroscience or neurobiology or biology even. It's actually expanded past that. So it's inspired a lot of the ways that we wire our robots, that we generate artificial intelligence. So what you're seeing here is a robot from Boston Dynamics that's practicing its walking, essentially. And a lot of the connectivity that was used to design the robot was actually inspired by central pattern generators in themselves. So that's kind of scary because they walk really well. You could actually shove them, and they can kind of learn to avoid your shoves. But yeah, so that was rhythm and tempo, and that's pretty much all I had for you guys today. And I hope you enjoyed at least listening to some of that. I talked about neurotechnology, my own research in birds, and this idea that the rhythms and larger scale activities, electrical activities of the brain matter a lot in the way you perceive the world, the way you perceive time, the way you act in the world, and the way that you chain together sequences and that you actually learn. So thanks a lot for letting me come to talk, and thanks a lot for listening. I'll take questions, and we should make it more of a conversation.